So there are four things that are detrimental to the origin of life, and one of those is oxygen. So if oxygen was around, you're not going to get this process of spontaneous generation of life. If it happens in water, whether it's a deep thermal vent or if it's just a little pond, you're not going to get polymerization of nucleic acids or of proteins because water breaks these compounds apart. If you have ultraviolet light or raw energy is destructive to all biologic processes. And if you have it in an uncontrolled way, it just breaks things down. It doesn't build. And then amino acids, remember we said that it has a specific design, a left-handed design as opposed to a right-handed design. These characteristics suggest that what Miller and Urey and others were doing was suggesting a scenario that would never come to fruition as a way to explain how life became life. And then, of course, there was the problem of the chicken or the egg. We've alluded to it before. DNA creates protein, and the information in DNA is pushed out through the nucleus in the RNA, and it creates, through transcription and translation outside of the nucleus, proteins. So proteins require DNA for their formation. But DNA requires protein for its replication. As you saw, polymerases are proteins, histone proteins are proteins, topoisomerases that open up the DNA structure are all proteins. So the question was, well, what came first? You need to have both to have either. Well, so what happened in about 1980 is they said, well, we're going to ignore that problem, and we're going to say RNA, not DNA or protein, was the first original substance. And so they take this ribonucleic acid and they say, well, this structure, a single-stranded RNA, can actually fold back on itself by Watson and Crick base pairing. And that molecule can actually store information in its strand. That's how it brings information from the DNA outside of the nucleus. And they found in the early 80s that these RNA molecules could function like an enzyme. And so the single-stranded RNA can actually cut itself without a protein enzyme. So they call it a code and a catalyst. And everybody was really excited in the early 80s and frankly for the last decades about RNA as being the foundation of a view of the world about how the world made itself. So it's an RNA world according to many people. And then of course you have interesting diagrams that people draw about. Well, you start out with RNA that begins to replicate, and then you have proteins coming from that RNA, since RNA is involved in making proteins, but somewhere you would have gotten ribosomes. But then on the other track, on the parallel track in B, there's the lipid track where the lipids kind of come together and organize themselves, and all of a sudden those two come together to form a living cell, and you have a gene portion and you have more of a metabolic portion. So you find that people are really separated into two camps. People who are the gene first people, the strict RNA people, and the people who say, well, genes are fine, but you got to have metabolism for life. You got to have something that's going to generate energy. And yes, you got to have both of them if you're going to have life. And you got to ask a lot of questions like, why would RNA make DNA in the first place? Why would it make a protein if it's perfectly fine about being catalytic and all these kinds of things? Why would it make metabolism? I mean, these are questions about a scenario that tend to get ignored, as well as where did all the information come from in the first place? So Jack Szostak, in a 2001 Scientific American article, said this. He basically said making life from RNA is easy. Now, recall that we started out about 1953, 1954 with George Wald, and now we're up to 2001. And he said a nucleotide, an RNA nucleotide, is made of three things: a sugar, a phosphate, and a base of some kind. And that's what's diagrammed there on the left-hand side. And it's easy to make sugars. All you do is take formaldehyde and you heat it up a little bit, boom, you get a sugar, reorganizes. The catch is that ribose sugars, which is the kind that RNA requires, the one with two hydroxyl groups down here, is really hard to make. And in fact, it breaks down in exactly the kind of alkaline solution that you were going to use to cook formaldehyde to make a sugar. You have to have phosphates. Now, phosphorus is the element. Phosphate is a PO4 group. It has 4 oxygens. Phosphorus is not soluble in water unless you heat it. And under pressure, and you got to do that. Otherwise, you don't get soluble phosphates. Another idea is that there are Schreibersite. Schreibersites, these are parts of meteors that have come in that actually can be dissolved in water and give phosphates. The third part, of course, is a nucleobase, a base of some kind, nitrogenous base. He says it can assemble spontaneously from cyanide and acetylene, 2 of the things that Miller created. Unfortunately, he doesn't know how to make cytosine, which is one of the key bases. Cytosine deaminates into uracil, so you can figure out how to make uracil. So he says, I can make all these three things in the laboratory, ribose, sugar, phosphate, and base. All I have to do is put them together, and boom, they'll form a nucleotide, right? Nope. Put those 3 things together, and they don't form anything. The reason is that water prevents that reaction. If you do it in water, it won't let it happen because of the mass balance of the equation. Hydrolysis keeps these apart. You don't get nucleotides forming in that situation. What you need is energy, and what you need is regulated energy, energy that's regulated through catalysis to click this onto that and to take that and click it onto that. It doesn't happen randomly. And the same thing is true of DNA. DNA is an imposed structure. You take nucleotide parts, you put them together in water, they don't form DNA. You need to have mediators, enzymatic mediators of energy compounds to make these structures. They are non-random structures. They are not chemically predicted from their components. Can't throw the components in water and magically get these things out. Well, so some of these problems have caused people to say, well, maybe we ought to look at something other than RNA. So in the last couple of decades, people have said, well, let's look at life coming from hydrothermal vents deep in the ocean. Because, hey, high pressure, high temperature, reducing environment, maybe we could get life starting there. And as a matter of fact, there's lots of life in some of those very deep fissures in the ocean. That is very thermostable, and bacteria are some of those bacteria are really strange and very tolerant to heat and pressure. And the idea was if this thermal vent has these little cavities in it, it'd be a great place for life to form in this little protected cavity. That hasn't worked out either, but people are certainly thinking about that idea. But now there are other groups. One group says, no, RNA was first, but it formed on ice crystals. So it wasn't hot and it wasn't deep in the ocean. It was dry, essentially, in ice crystals, frozen water. So it's an ice RNA world. Another group says, no, it was hot mineral RNA. So you take minerals as the surface of minerals in a hot environment, and that can take these different parts and stick them together to polymerize them, these different nucleotides, et cetera. And that can happen on the surface of minerals. The third group says, no, it's a medium temperature environment. And it's mostly made of zinc. In other words, it's an ion catalyzed kind of a world. Okay, so why are we going on to these kinds of ideas? Because URI didn't work out, because RNA didn't work out. Now we've got cold RNA and hot RNA and zinc RNA. Why are we going on to all these models? The reason is we haven't found any answers, only more questions. You can make parts of things, but then there's ad nauseum caveats and problems in the chemistry of how this would occur. And it's very, very frustrating. This kind of persistence on luck, on time and chance, leads to misconceptions of reality, doesn't it? It leads to blind alleys. It leads to an endless search for answers. Whereas a focus on something that has intent can lead to you in a search for truth, a resolution of some of these questions, a rationale and an explanation that's actually satisfying for the things that we do observe, not for the things that we can't explain. So these people are persistent in the face of failure for, 90 years now, depending on what time frame you take. And the reason is they are committed to philosophic naturalism. But they say, Robert Hazen said, no one knows how life arose on a desolate young earth. But you can be sure it was a chemical event. It wasn't a designed, purposeful event. It was just a chemical event. Life was was inevitable as was the whole of organic evolution, says George Wald. It lies within the order of nature, and apart from the details, it was inevitable. They're committed to that proposition. Many people were committed to the proposition that the early atmosphere had no oxygen. Committed to it, because that's the only way that they could have any precursors to life by scientific means. So what does this all mean? It really means that as you look at this data, there's really no evidence for an early organic soup. And such a soup would have been too dilute in any case to form any meaningful molecules. And besides, with all the destructive forces in there, that would have never happened. And so there's actually no hard evidence that such a soup ever existed. It's simply hypothetical. And that oceans or ponds would be the last place that you would think life would start on a chemical basis, because water drives those components of life away. And there has to be something to overcome that. Now, you can force it, to be sure. We do this in the chemistry lab all the time. Organic chemistry lab excludes oxygen, excludes water, it creates temperatures, it adds enzyme facilitators, catalytic surfaces, and certainly chemistry that we use in industry does the same thing to make products under high pressures and temperature. You can force things to happen, but it doesn't happen naturally. And so there's no consensus on a model for precellular life despite 90 years of work. And the question that's always avoided is that question of DNA. Where does all the information come from? Not only do you have to account for the atoms, the elements, the molecules, the polymerization of all the molecules, the persistence of those molecules, the replication of those molecules, all of this, where did the information come from that they carry? And this is a quote from Werner Gitt in Germany. Since the findings of James Watson and Francis Crick, who solved the DNA structure and the Watson and Crick base pairing, it was increasingly realized by contemporary researchers that the information residing in cells is of crucial importance to the existence of life. Okay? Anyone who wants to make a meaningful statement about the origin of life would be forced to explain how the information originated. All evolutionary views are fundamentally unable to answer this crucial question, including the evolution of life, the chemical evolution of life. And so what you see here is a table of the genetic code, of codons, the three-letter codes that identify a particular amino acid for a sequence in a polypeptide chain as part of a protein molecule. And you studied this in general biology, but codes are languages. Languages are agreements between intelligences. Codes don't come from nothing. They don't come from random throwing of the dice. Codes come from an agreement about what something means. And so not only does the DNA have to agree with the RNA about what this three-letter sequence means, but when it goes out into the nucleus, there's something called a transfer RNA that takes the amino acid and brings it over to that spot. Remember, you saw that bringing in that three-pronged transfer RNA? That has to also agree that I'm going to take glycine and fit it in at this particular setting. So nucleic acids and proteins have to agree on the language. That's a level of complexity that doesn't happen by chance. And then you think about the amount of information and instructions in the DNA, in these three-letter sequences that line up amino acids. And then the proteins have functions in metabolic pathways that are finely regulated. Proteins are structures, proteins are enzymes. You see, the ink did not write the book. And I want you to think about that. Because as you look at a book or you look at the ink on a page, what we're basically saying is that the chemical properties of the ink and the paper wrote the information in the book without an author. That's what we're really saying when we say life designed itself, life built itself. Life doesn't need an outside intelligent designer. So, are you really ready to say that? That the ink in every book that you have wrote the content of your book? Well, we know that's not true. An author wrote the book. The question is, who's the author? We think it's the God of the Bible. For by him were all things created. That was Christ. All things were created that are in heaven and that are in earth, visible and invisible. Whether they be thrones or dominions or principalities or powers, all things were created by him and for him. God is the source of life. He created matter and energy for a purpose. He created time to allow that matter and energy to work and create life. He's the source of the information. He's the one that wrote the information on the chemicals. It was God who is the author. So the question is, can life make itself from nothing? I would suggest to you the answer is no.